Good morning, student. We will continue the topic chemistry of lipids, competency number 4.1, and today's topic is classification of fatty acids, their nomenclature, numbering, functions, and biological importance. So first we will see what is fatty acid. Fatty acids are defined as hydrocarbon chain with carboxylic acid group. Here carboxylic acid group is the functional group. Fatty acids are classified in three different ways. First classification is depending upon the number of carbon atoms present in them. Second classification is depending upon the length of hydrocarbon chain, and third classification based on presence or absence of double bonds in the hydrocarbon chain of fatty acids. So first classification, depending upon the total number of carbon atoms present, fatty acids are classified as even chain fatty acids and odd chain fatty acids. Even chain fatty acids have even number of carbon atoms. Example, palmitic acid with 16 carbon, butyric acid with 4 carbon. Odd chain fatty acids have odd number of carbon atoms in the hydrocarbon chain. Example is propionic acid. Depending upon the length of hydrocarbon chain, fatty acids are also classified as short chain fatty acid, medium chain fatty acids, and long chain fatty acids. Short chain fatty acids has carbon atoms between 2 to 6. Example, butyric acid. Medium chain fatty acids have carbon atoms ranging from 8 to 12. Example, lauric acid with 12 carbon. And long chain fatty acids have 16 or more than 16 carbon atoms. Example, palmitic acid. Here we have classified only even chain fatty acids. Third type of classification is depending upon the presence of double bond. Saturated fatty acids and unsaturated fatty acids. Saturated fatty acid does not have double bond in the hydrocarbon chain. Example, palmitic acid with 16 carbon and stearic acid with 18 carbon. Unsaturated fatty acid means they possess one or more double bonds in the hydrocarbon chain. Unsaturated fatty acids are further classified on the basis of number of double bonds present in them as monounsaturated fatty acid with one double bond. Example, oleic acid. Polyunsaturated fatty acid with more than one double bond. Example, linoleic acid, 2 double bonds, and linolenic acid with 3 double bonds. Coming to the nomenclature of fatty acids. Here the carbon atoms present in the fatty acids are named in different ways. Sometimes they are named as alpha, beta, gamma, delta, epsilon carbon, starting from the carboxyl end. Here alpha carbon is that carbon which is adjacent to the carboxylic acid group. Second way of naming the carbon atoms of the fatty acids are by giving the numbers to the carbon atoms as 1, 2, 3, 4, 5, etc. Here the number 1 carbon atom belongs to the carboxylic acid group. Third way of classification is by giving number omega 1, omega 2, omega 3, and here the omega 1, omega 2, omega 3 starts from methyl end of the fatty acid end. That methyl end is also called as omega end. Next is omega 3 fatty acids and omega 6 fatty acids. Omega 3 fatty acids, these are the fatty acids in which the first double bond from the methyl end is present between omega 3 and omega 4 carbon atom. Example, linoleic acid. Omega 6 fatty acid means the fatty acids in which the first double bond from methyl end is present between omega 6 and omega 7 carbon atoms. Example, linoleic acid and arachidonic acid. Now come to the polyunsaturated fatty acid. So Polyunsaturated fatty acid contains 2 or more double bonds. They are mainly found in vegetable oils and fish oils. Examples of these polyunsaturated fatty acids are essential fatty acids, arachidonic acid and docosahexaenoic acid. We will study these 3 types of important polyunsaturated fatty acid in details. First, essential fatty acids. As the name suggests, they are essential for the body but not synthesized by the body. Examples: linoleic acid and linolenic acid. Linoleic acid having 18 carbon and 2 double bonds. It belongs to omega 6 family, and linolenic acid is also having 18 carbons but 3 double bonds, and it belongs to omega 3 family. Here is the figure of linolenic acid and linoleic acid. First double bond between omega 3 and omega 4 belongs to omega 3. When the first double bond is present between omega 6 and omega 7 from methylene, then it is linoleic acid. Coming to the sources of essential fatty acid, linoleic acid is found in corn oil, cottonseed oil, egg yolk, peanut oil, soybean oil. These are the rich sources of linoleic acid. Then linolenic acid, it is mainly found in linseed oil, soybean oil, cod liver oil and fish viscera. Now we will study the biological importance of essential fatty acid. First, these essential fatty acids are important components of cell membrane. All of us know that cell membrane is made up of phospholipid bilayer. In that phospholipid, at second carbon atom, unsaturated fatty acid is present, and it is mainly the essential fatty acid. Essential fatty acids also have important role in reproduction. This essential fatty acid decreases serum cholesterol level. These essential fatty acids are important structural components of HDL, which is a good cholesterol or which decreases the serum cholesterol. Essential fatty acids also act as a lipotropic factor. Lipotropic factor means these are the substances which prevents fatty liver. Fatty liver means excessive deposition of triglycerides in the liver, leading to liver damage. Essential fatty acids are also important components of mitochondrial membrane, and these essential fatty acids are required for optimal vision. In case of deficiency of essential fatty acid, the optimal vision is diminished. The double bond present in the polyunsaturated fatty acid is having cis configuration, and due to this there is bending in the chain of essential fatty acid. The diagrammatic representation is shown in the tail. There is some bending, and due to this bending in the essential fatty acid of the phospholipids, when these phospholipids forms cell membrane, these phospholipids cannot tightly packed because of this bending in the chain of fatty acid, and this will increases the fluidity of the cell membrane. If the phospholipids are tightly packed, then cell membrane will loses its fluidity. So it is the essential fatty acids plays an important role in maintaining the fluidity of the cell membrane. As the essential fatty acids are integral part of mitochondrial membrane, in the deficiency of essential fatty acid, biological oxidation is reduced. Now coming to the next polyunsaturated fatty acid called as arachidonic acid. Arachidonic acid can be synthesized by the body if we supply linoleic acid in the diet in sufficient quantity. This arachidonic acid can be converted into prostaglandins, thromboxane and leukotrienes, which are very important biomolecules in the body. Arachidonic acid forms 10 to 15% of the fatty acids present in the membrane. Next, docosahexaenoic acid. It is also synthesized from linolenic acid. It is present in high concentration in fish oil. In our body, this docosahexaenoic acid is found in large concentration in retina, cerebral cortex and sperms. The levels of these fatty acids are found low in patients with retinitis pigmentosa. These polyunsaturated fatty acids, due to presence of double bond in them, they are easily attacked by the free radicals or the enzyme peroxidase, leading to the oxidation of this polyunsaturated fatty acid called as peroxidation. When there is peroxidation in the fatty acid present in the cell membrane, it may damage the cell membrane. Elevated level of polyunsaturated fatty acids are seen in Zellweger syndrome. Now come to saturated fatty acids. Fatty acids with no double bond are saturated fatty acid. There are so many examples of these fatty acids are there, but we will see only two important examples. One is trans fatty acid and another is palmitic acid. Before that, we will see what is meant by hydrogenation. Hydrogenation is the process by which we will add hydrogen atoms to the unsaturated fatty acid and convert them into saturated fat. Here I have given one example of linolenic acid, which has 3 double bond. When we add 2 hydrogen atoms, it gets converted into another 18 carbon linoleic acid with 2 double bonds. When we add 2 more hydrogen atoms, then it gets converted into 18 carbon oleic acid with single bond. And when we add again the hydrogen atoms in it, it forms saturated fatty acid called stearic acid. Hydrogenation of oils may lead to solidification to fat. Now, trans fatty acids. Trans fatty acids are generally produced by partial hydrogenation of cheap vegetable oils. Why these are prepared? Because just I have told you, in the vegetable oils mainly the unsaturated fatty acids are present. These unsaturated fatty acids are easily oxidized by free radicals and peroxidase, which will impart unpleasant taste and smell to the vegetable oil. To prevent that oxidation of unsaturated fatty acid, this unsaturated fatty acid can be converted into saturated fatty acid by hydrogenation, so that their shelf life is increased. Now, the dietary sources of these trans fatty acids are dairy products, fast foods, bakery products and processed foods. Trans fatty acids are injurious to the health. Firstly, they compete with the essential fatty acid present in the diet for absorption, leading to decreased absorption of essential fatty acid from the diet. Therefore, they increases the deficiency of essential fatty acids. When we consume large amount of trans fatty acids in the diet, they get incorporated in the cell membrane, due to which the fluidity of the cell membrane is lost. Trans fatty acids are also found to be harmful in cases of cardiovascular disease and atherosclerosis and diabetes mellitus. In this case, they increases the plasma lipids and LDL lipoprotein, which is a bad lipoprotein. They decreases HDL lipoprotein. They causes systemic inflammation, particularly in the blood vessels, leading to damage to the endothelial cell. They also causes endothelial dysfunction, insulin resistance and obesity. Next is saturated fatty acid. The most common is palmitic acid with 16 carbon. It is the main fatty acid which is synthesized by the body. It is the fatty acid which is stored in the adipose tissue in the form of triglyceride. This palmitic acid undergoes oxidation in the fasting state to provide us energy. So this is the structure of palmitic acid. Here actually it is a structure of triglyceride called as tripalmitin. Glycerol molecule is there, to which 3-palmitic acid is attached. And this completes our topic of fatty acids. Thank you very much.